always you are asking, right? So let's explain, or let's try to find out the mechanism behind persistence. Also, towards the end of my lecture today, I'm going to talk part of our research data to show you how we study the diseases. Okay, so persistence of pathogens. Factors responsible persistence. So again, we are telling the same story in different ways. We mentioned a little bit about viable but viable but non-culturable state. We'll examine a little bit that state of so these type of cells or cell population, they also maintain a called viable but non-culturable state, and that also are a community of persisters. Okay, I tried to explain that last time. What is viable but non-culturable? Then biofilm formation, obviously, those cells try to form biofilm, or within the biofilm community, they are also persisters, right? Quorum sensing, of course, that plays an important role, and spore formation. That's also a strategy for microbes to serve, maintained as a persister. Of course, we are not going to talk much about the spore formation, but that's a natural process for some of those microbes like Bacillus or Clostridium. Many of these properties actually start with stress, right? Biofilm is a stressful condition, or bacteria going to a persister phase, that's because of the stress. Okay, so that's why we're going to spend a little bit of time. What are the stress means to you? I mean, you can apply that information to yourself. So if you are under stress, are you more productive or less productive? A lot of times, negative effect, right? You become less productive. So that means you go through some kind of dormancy, or kind of like a less active phase. So some of those stress can make the organisms going through that transition to become more persistent. So we're going to examine that. So here we're going to talk about stress response and adaptation, and then effect of pH, salt, acid, temperature, any of those standard, physical parameters that can impact the cellular physiology that leads to persister. And I mentioned a little bit about viruses last time. So viable but non-culturable state. Let's look at those populations. Organisms that are no longer culturable with standard non-selective culture media. So this terminology VBNC is critical for microorganisms. As I mentioned yesterday, that you take a sample, you try to grow them in the culture media, and then you find out there is no growth, right? If you change the condition, then after some time, then you start growing. Means that same sample behave differently. So that's called the viable but non-culturable. So what happens? Their metabolic activity, the metabolic process slows down, or they may be producing certain enzymes that's needed for their to maintain their viable but non-culturable state. Maybe they are not synthesizing enzyme, or they need enzyme that's required for harvesting energy so that they could be metabolically active. But in this VBNC state, some of those functions may be arrested or stopped or halted or not at all. Everybody got it? So some of those enzymatic activity slows down or stops. Nutrient uptake, they may not be taking uptake nutrient, therefore that also slows the metabolic process. And other aspect, also a lot of times the cell change their morphology. Okay, that's kind of fascinating. The cells, they could be rod-shaped cells, and then suddenly they become round cells. Okay, but we are so much trained to study microorganisms under microscope. Like we know some organisms are always rod cells, right? Or some cells are always round cells or small cocci. Okay, so that's our knowledge, that's our familiarity. But as soon as you see certain cells change their morphology, then you get already, I said, oh, it doesn't look like it's a salmonella, or it doesn't look like it's E. coli or listeria, because the shape changes. That I'll talk about that. And so those are some of those attributes that relate to the viable but non-culturable state. So some of those examples of microorganisms which goes through BBNC state: Campylobacter, E. coli, Listeria, Salmonella, Shigella, Vibrio. That's original study actually started with the Vibrio. So let's look at a little bit about the stress response. This looks a little bit complex. Let me simplify it. So there is this called level of stress. There is a degree of stress, right? Some stress could be high level of stress, right? We all know that. Or some could be moderate, or some could be low level stress. So depending on the degree of stress, you may see different effect on the bacteria. So let's say this is a normal growth range. This is the I want you to pay attention to the left side of the graph. So this is the level of stress. Some stress could be sublethal, means it's not killing it, but it keep it sublethal. They can regenerate or come back. And some of the lethal means they are going to die, right? We don't worry about that. You introduce some stress. So for example, heat, right? You started heating bacteria or exposing them to 50 degrees, for example, 50 degrees C. So you will see that some cells, it's going to be lethal to them, but some bacteria, it may not be lethal. So that will be sublethal. Everybody clear? So under 50 degree, which is not normal for that bacteria, so they are going to go through stress. And so what happens in this sublethal, if it is that 50 degree, so some cells will have an injury, okay? The cells will be injured, but they are not dead, but injured, okay? And then there is some stress adaptation. If it is a low level stress, not sublethal, there will be some level of adaptation. And we always know that we can adapt, right? Even the temperature going up here, we can adapt. We are not dying, right? But we are highly stressed. You cannot function properly, right? You cannot think properly if you are under stress. So that's where we can adapt. But when sublethal, that injury, this can be repaired. It's not that we are irreparable. We can repair ourselves. So this is a sublethal injury. But if you look at this window between lethal and sublethal, and this is where the bacteria can get into non-culturable state. So it's not dead, but they are like a unresponsive. It's like a totally dormant. Are you getting it? It's like a zombie. You've seen these zombie movies, right? So the dead coming back to life. So they are staying dead all the time, and suddenly they become alive. And so kind of like they fall in the grand scheme of stress response. And of course, like I said, the dead cells, you cannot culture them. They are always dead. They are not going to be recovered. So just give you that idea. Now let's look at it. What is this persister terminology and persister phenomenon came? So the BBNC states, like in this plot, if you look at the bacterial cells, if there is environmental stress, and these organisms are going to be part of this stressful community, then when they are under stress, some population can start forming biofilm. Now start thinking the reverse way. If there is a stress there, and that may promote bacterial biofilm formation, and then some population within that, okay, they would be called persister cells. Means they are not responding, they are kind of dormant. They could be BBNC cells, okay, and they are just become persister. So why that happens? Nutrient limitation is one factor. Changes in sodium chloride, levels of sodium chloride in the water, especially in the ocean water. That's where we see a lot of BBNC cells. Exposure to hypochlorite, shifts in temperature. So a lot of these factors contribute to the BBNC state. Think about it now, that when microorganisms, you are trying to kill them by a number of different ways, and some of these happen to be some of those factors. So obviously, we can create some of those organisms to undergo VBNC state. That makes sense? Because we are trying to kill them, right? We are going to induce injury, then followed by lethal treatment. But when we are not perfect in providing that environment, or even in the nature, that can happen. And so the organisms can go through VBNC state. So this one is just saying that VBNC cells, we can resuscitate them. Then you have to reverse it, right? They are not dead. They are dormant. They are staying in a place. They are not responding to natural conditions. But if you shift the temperature, okay, so normal temperature is 37, and if you shift it to say lower direction, 30 degree, maybe that's favorable for that organism. Now it can wake up, can come back to life. Maybe that can grow. Now they can reverse back. Also, return to nutrients. So if there is a nutrition starvation, now if you give them food, they can come back to life. So how about the catalase? Sometimes we produce hydrogen peroxide. Like for example, in the macrophage, when the bacteria are engulfed, macrophage produces hydrogen peroxide. And hydrogen peroxide essentially going to kill the organism, right? But if you make catalase, you can neutralize the hydrogen peroxide, okay? So that means if we add catalase, now you can neutralize hydrogen peroxide action, now the organism can come back, okay? And sodium pyruvate also, it scavenges some reactive oxygen species, so the organism can come back to life. So these are some of those factors you have to add it to the media, and then if you have a VBNC cells, they can come back to life. And that's essentially what we do. So in order to avoid being VBNC state, you can resuscitate. Everybody understand the resuscitation? Resuscitation might, like if you suddenly lost your consciousness, right? And then or you were dying, right? Or you had a heart attack, and so your heart stopped. And so people start to resuscitate them, right? They give mouth-to-mouth breathing, right? They do the whatever called the heart pressure, right? To bring the heart back. And that's called resuscitation in real life. And so the organisms which undergo stress, undergo injury, you need to bring them back from VBNC state or injured state to healthy state. And that's what we call resuscitation. And so resuscitation state needs to be followed through. Okay, so in this one, if you have active cells, we know they are going to multiply, right? If you have a persister cells, those who are dormant, those who are not responding to any nutrient, you need to bring them to life, right? So that's why there is a step in between dotted line. We need to return to the active condition. If you bring them to active state, now they are going to multiply. You can culture them, right? So this is, then if you have a VBNC state, VBNC also part of the persister community, right? And so now you can make them to persister cell and then become active, and then they start to grow or multiply. So all of these factors you have to understand how to bring them back to normal conditions, so then you can grow them. Okay. So persister cell, you can easily study it. I think there are a lot of these nice review articles came out. So if you take a population and expose to antibiotic, for example, right? And you know that as soon as you expose to antibiotics, those who are resistant, they are always going to grow in that condition, right? So let's take that population right here. Then those who are susceptible, they are going to die right away, right? But there is a population which is not going to die, but this is a tolerant. Tolerant also a little bit different. They are not resistant, but they are tolerant. They can tolerate up to a certain level. Okay, but eventually, as you increase the concentration, they are going to die. Tolerant is all dependent on the concentration of the antimicrobial agents or the antibiotics. But there is this population after antibiotic treatment, they are not tolerant cells, they are not susceptible, they are not resistant, but they are persistent. And that happens when you treat the patient with antibiotics, and these persistent cells can eventually come back. Very clear? And this is what we've seen. They are not resistant population. So something happens to this population of microorganisms or bacteria. They tend to convert into called persistent cells. And so to understand the mechanism of persistence, you have to really understand the molecular control, the regulation. Okay, I'll try to explain you a little bit, but probably it needs a lot more reading, but try to simplify it. So persistent cells, this has been a long history. Okay, for I think 1942, that's when the Hobby actually he discovered the phenomenon. This individual, Joseph Bieger, actually named these small numbers of bacteria that survive penicillin treatment, and those are called persisters. The actual terminology coined from 1944. And then over the years, there's a lot of work being done, and they're trying to understand the mechanism. And this actually came from this article, review article, published actually 2024, and talked about it. So I try to simplify it. This is really a complex structure, and why certain cells undergo persistent stage, and how do they do that? Okay, why this happens? So there is two phenomena. We call it toxin antitoxin phenomenon. Okay, in cellular system or microbial cells, the toxin antitoxin works cooperatively. Okay, when you say toxin, means it's going to degrade something. Toxin has a negative effect, right? That's the simple way I try to present it. So if you take a toxin, it has a protease activity, it's going to degrade messenger RNA, for example, right? And so there won't be any protein synthesis. So if you don't have that protein synthesis, and that protein is essential for your survival. So let's say protein important for substrate utilization or enzyme. Okay, if that's not produced, then what do? You cannot use that enzyme to harvest energy. So therefore, in that case, your system not going to work. Means it's going to slow down. Then antitoxin is kind of like a balance. It has some regulatory element, and so this antitoxin toxin effect always kind of working together. Okay, so toxin is going to try to degrade it to make it cells like a persistent. I don't have enzyme, so I cannot use the substrate. I'm not dead, but I can maintain my life. But antitoxin, when it comes, it will contradict that. And as soon as that antitoxin starts working, then the persistent cell can become more healthy cell. Okay, that's the simple way I can present it. But it's all enzymatic action, how they manipulate their messenger RNA for production of say enzyme or toxin, which are essential for their survival. Got it? So I want you to read it. It's a little bit complex phenomenon. This is a nice review article, talked about how this works. Okay, other central molecule play important role. So persistent cell formation by toxin antitoxin system. So if you say that genetic basis of persistent formation, this toxin antitoxin I just mentioned, that helps maintain the dormancy state and controls the cellular process. Okay, you are going to get this slide, so obviously you'll be able to read it up more. And then there are different types of inhibition happens: type 1, type 3, then type 2, and then type 4. These are all called antitoxin toxin effect, and that's how it mechanistically helps the cells to maintain persistent state. Okay, this is the simple way I try to present it. Okay, so strategies to control persistence. How do you control these persistent cells? Obviously, they are a problem, right? You try to kill them with antibiotics. Some of them are not killed, but then they are still maintained, right? So how do you manage them, control them? And so this review article, again, it really took nice approach to say, okay, you need to have a combination therapy, right? So for example, you need to have a anti-persistent drugs together with antibiotics. So you can just say, okay, I can give you a penicillin, but penicillin may not kill that cell, right, alone, which is going to maintain a persistent stage. But then can I have anti-persistent drugs together with the antibiotic? Then you will be able to remove all those cells. Here they used antibiotics, and then some drugs, again same mechanism, and then drugs are sensitive to persistent. Immunomodulators. So there are a number of, you can use, say membrane damage component, which can help the drug to penetrate. through the cells which are resisting certain antibiotics. Okay? So all of these combinations can be used to help make the cells a persister. So again, I'll try to give you some idea, but you need to read up a little bit more because it's not possible to explain everything within this short period of time. Just trying to give you an idea so that when you go and get a chance, read up some of this. Okay, so as I said, stress become one of the major factor in inducing the cellular process. Okay, whether it's a VBNC state or persister state. So these are the common factors, right? We talked about temperature, acid, antimicrobials. Then of course the biofilm. When the cells form biofilm, part of the community become persister. Okay, so cell morphology, like I mentioned about that, the cells change their morphology. Look at the vibrio, how they look like. They are curvy rod, short rod. Okay, that's how the vibrio look like. This is a vibrio cholera on the left. But when they get into VBNC state, this is how they look like, round cells. Have you ever thought that rod-shaped cells become round or coccoid? And so when you look under, we did some work on this a way back, and we looked at the VBNC vibrio, they are round cells under microscope. So when you look at it, then you said, no, it might be something else. You never think that this is vibrio. But if you reverse the condition, so ideal environment, and that's what become a rod-shaped cell. So when you are testing the water, say for example seawater, that's where you see a lot of vibrio cells, and you bring the water into the lab and then do the microscopy, you might find this type of cells. This is the majority population. Then this become rod-shaped cells, and you can reverse that. You can induce VBNC state, and then rod-shaped cells become round circular cells. Okay, so those sort of things you can manipulate. Okay, what about this? So you can see these small cells, the shorter rods, right? What is this cell? If you are looking under microscope, what would be your first impression? It's a contamination, right? As a microbiologist, always you used to see certain uniform population. If you see a different shape, size in same population, you immediately think, oh, this must be something else. So actually, this is salmonella. Okay, these are salmonella cells, and some salmonella decided not to divide. So this become a long rod cell. If I have seen a bacillus under microscope, bacillus make a long chain. Okay. Now salmonella decided to become a long salmonella cell. Very unreal, right? And you will think that immediately it's actually a contamination. Okay, now answer my question. Yesterday I discussed about cell wall, right? I talked about cell division a little bit. So what do you think is happening in these cells? Why this cell became a long rod-shaped cell? When we see single cell, what does it mean? Single cell become two cells, right? And they separate from each other. So then we see these two single cells, right? And this is how they divide. That's their natural process. So during cell division, what happens? I'm looking at, so yesterday remember peptidoglycan structure I was showing you, and I showed you N-acetylmuramic acid, N-acetylglucosamine, how they are linked and how they have cleaved. There are enzymes, right? Well, there is a peptide there, peptide bridge, but between N-acetylmuramic acid, NAM, NAG, there one beta link, right? And did I mention some of those enzymes which cleaves those? I was using Staph aureus as an example. So what is happening with this stress? So those enzymes are not working. They make a lot of those autolytic enzymes. That's what helped them to divide. They become a single cell. So if your stress inhibit those enzyme synthesis, cells are not going to divide, right? So apparently this long cell, somehow some of those enzymes are not working. Stress can do that, right? Stress can, that's the whole idea. The stress factor can control the expression of some of the gene products which are responsible for, in this case, cell division. Got it? Everybody clear on that? And so look at this. Filamentation caused by starvation. What did I say? Nutrition, right? Lack of nutrient. That can be long cells. Low temperature, low water activity, desiccation, preservatives. A lot of those factors can induce stress, and so the bacteria would behave differently. Everybody clear so far? Can you link the dots I've been trying to address? Okay, let's look at another organism. This is Listeria, which is very short rod cells, 1 to 1.5 micron long. And if you hit them, can you see this? These cells are long. And what else they are doing? Aggregating. Look at that when they grow them at salt, 5.5%. If anyone looking at these cells, you will never think these are Listeria, right? Actually, we are used to seeing Listeria like this short rod. But if you grow them under 5.5% sodium chloride, which is pretty high, very high, okay, and this changes morphology and the shape. And if you look under that electron microscopy, look at this short rod, and now you can see these cells are joined, and look at this salt. I never seen Listeria become such a long chain. What happened again? Salt interrupting this enzyme activity, which is supposed to break it, separate the cells. That's not happening. So these are very common phenomena. This is just recently, we are working with E. coli, it's enterotoxigenic E. coli, and send this culture to my colleague, and we are trying to see their responsiveness to antibiotics. Okay? And so he said, well, let me grow it in the ampicillin because they could be ampicillin resistant. Have you ever thought this is E. coli? And then as soon as you remove the ampicillin, look at this. It reverts back. So you need to be preparing your mind that any time you change that condition to stress can change the morphology. Again, it's arresting a lot of those enzymatic action, and then cells become totally morphologically different. So that gives you a clue that why they are not going through BBNC or persister phase. Okay? But morphology is pretty dramatic. I mean, you can clearly see there is no question about it. Okay? All right, so that changes. Now, I think of what I was trying to tell you about the role of, let's see. Okay. So next I'm going to look at example, tunicamycin. Let's use one antibiotic. Let's play with it. Okay? So antibiotic we add at concentration to inhibit microorganisms, right? Everybody clear? Okay, so what if we don't provide the actual lethal concentration? Lethal concentration means concentration that's going to kill all the organisms, right? But what if I provide sublethal dose? What happens to the microbes? Right? Because that happens, right? Sometimes we don't take proper dosing. And so does it affect the microbial behavior? So I showed you this yesterday, right? And how the peptidoglycan synthesis works, and then how the teichoic acid or wall teichoic acid synthesis. These are the two major gene products, right? These two genes help in the peptidoglycan synthesis, and taro is actually for wall teichoic acid synthesis. So tunicamycin, which is a small the antibiotic, it inhibits actually this gene. So therefore, this teichoic acid synthesis is inhibited. Okay, and I mentioned that wall teichoic acid also play an important role in biofilm formation, right? So now, if I don't kill the organism, because if I kill the organism, I cannot study its effect, right? So I need to give a sublethal dose. So that's what I am trying to explain to you. Okay, let's say I gave a 2 low doses of tunicamycin to the bacteria, and see how that affects the microorganism. First of all, we use Staph aureus and Listeria. So this is zero amount of tunicamycin is control. If you look at the cell morphology, it's still maintaining round shape, and you can see the cell wall and cytoplasmic membrane pretty clear. Okay. As soon as you add a low concentration of tunicamycin, 5 microgram, we consider as a sublethal, you can clearly see the cell wall structure started to disrupt, right? You can see the peptidoglycan structure is very thin, and that says totally different, right? So it's working in a partial level. So structurally it's begin to affect. And then if you look at the same happen with the Listeria monocytogenes, 0 versus 5, and you can clearly see the cell wall structure becoming very unstable, very disrupted, right? And so it affects what we propose. Now I want to see how that affect the morphology. Tunicamycin at low concentration. So this is cell morphology for Listeria. You can see it's normal, and as soon as you use 0.3 microgram level, and you can see this morphology begin to change. A little bit higher, you can see also again change, right? So you are seeing that bacteria are undergoing stress, and therefore their morphology is affected. Already I mentioned that it's going to affect the morphology. How about the colony? If you look at the normal colony, and as soon as you have antibiotics, the colony morphology also changes. There is one type of technology we developed called light scattering, and this is a healthy colony, and it creates this beautiful scatter signature. But as soon as you have a colony formation like this, the signature different. So it's telling you that cellular morphology, as it affects being affected by antibiotic and its structure, and then its phenotypes also changes. Same thing happened with Listeria monocytogenes. Same thing happened with Staph aureus. Okay, this phenotype change, and you can see the cellular presence with the different concentration of antibiotic. What about the biofilm? Right? We mentioned that teichoic acid or wild teichoic acid is responsible for biofilm formation. Right? So does it affect biofilm formation? Let's look at it. You can ask a number of ways. So in this case, we are just counting bacterial cells, and this is the crystal violet staining. I think we are going to talk about crystal violet tomorrow for the students. And so if you have a antibiotic, 2 concentration, you can see that biofilm cells are reduced level. Biofilm here also reduced. And I mentioned this morning about eDNA. You can measure eDNA. eDNA synthesis also significantly decreased for antibiotics. So that means biofilm formation is affected. Just look at the images. Here, if you have a normal biofilm produced by Staph aureus, as soon as you have antibiotic, you can see it's dispersed and much lesser biofilm. So it clearly says tunicamycin affects wild teichoic acid, also peptidoglycan structure, and that also in turn affect the biofilm formation. So that means if at sublethal dose, if you are unable to kill them, you are going through these stressful conditions where microorganisms behave differently, they respond differently. Everybody clear on this? Okay. Then if you look at their ability to adhere to cause infection, I mentioned this morning again that adhesion is one of the characteristics, right? The microorganisms produce protein or carbohydrate to help them attach to surface, so that part of the infection model, and then they can cause infection. So if you have antibiotic at different concentration, their adhesion to cells, host cells, epithelial cells are significantly decreased, right? Same way with the Staph aureus, and while they are less toxic to those cells. And without going too detail, also we saw same phenomenon for Listeria monocytogenes. So the bottom line of this is that if the bacteria are under stress because of sublethal antibiotics, their infectious properties also changes, and also their biofilm characteristics. Okay, number of ways you can prove this, whether this affecting their virulence or pathogenesis. This is another way you can see if they regulate certain inflammatory pathway, and obviously you will see a decreased And this is cytokine production. Like I said this morning, inflammatory cytokines TNF alpha, IL-6, their level also decreased significantly because they are not infecting at that level. But they are still pathogenic, but their degree of pathogenesis is decreased significantly. This is also for Staph aureus, this for monocytogenes. So just trying to point out that the structural components which are integral to the bacterial properties that help them to form biofilm and also contribute to pathogenesis. And if I used a high concentration of tunicamycin, would have killed the cells, right? Then I will not be able to study all of those things. So you can design your experiment to induce stress and find out stressful conditions still make an impact on the microorganism's ability to cause disease or infection, at the same time biofilm formation. Everybody clear on this? Okay. Let's look at other factors. That's some studies. And if the organisms undergo stress, I mentioned that sometimes they don't produce certain proteins. So this is a clear example. Very simple example though. If you take Listeria monocytogenes and all of these media, I'm not going to name all of them, pressure broth, UVM, all of this, all this media has certain antibiotics. Okay? Different levels, different antimicrobial agents. Everybody clear? Now I grow them. Let's look at their protein profile. If you grow them, same bacteria in one media, I found only one and these two major proteins showed up. Same bacteria grown in another one, I saw this protein and this protein. Another media, you can see this profile. And this is another media. So this is fascinating, is that bacteria, because of different antimicrobial component, they stop producing certain proteins. Maybe some of those proteins are important for adhesion or attachment or biological function. But just to point out, when the microorganisms are under stress, they stop making certain proteins. Everybody clear on this? Any questions so far? Okay. This is another example. In certain media, they will make a lot of proteins. You take Listeria monocytogenes. In certain media, they will make a lot of proteins. In another media, oops, just forgot to produce proteins. If you are trying to detect this organism with this antibody, you are going to find negative. Or for running function. Some of them just never found any of these proteins. So the bottom line is that the stress can do a lot of different things to the cells. And no wonder they go through this type of transition. Okay, the diseases. Let's talk about the diseases caused by bacterial biofilms. I already showed you this slide before, right? You recall? And atherosclerosis, infection, endocarditis, all kinds of wound infection, cystic fibrosis, urinary tract infection, all kinds of diseases, right? We know that's responsible by biofilms, right? Now, this is also, you have seen this, dental plaque, mastitis, intestinal colonization, and so on. So, medical devices also you'll find this biofilm formation, right? And all of those we discussed. So, the wound infection, mastitis, we talked about it, right? That different types of organisms. So, these are responsible for causing diseases. So, biofilm also in the gut and the disease production, which is very important. Before I talk about the gut, the role of biofilm in diseases in the gut, first of all, one thing I want you to know, the normal condition. The normal condition is what? What you are, right? So if you look at the normal or eubiotic conditions, and you will see this epithelial lining, and then there will be a thick mucus layer, and a lot of microorganisms, healthy community, would be on that cellular location. So thick mucus, we call it tight junctions, diverse microflora, and these are all good properties. But as soon as just the opposite of the eubiosis, which is called dysbiosis, which is the abnormal condition. Maybe you are under stress or some infection agents. And so what happens in these biotic conditions, sometimes this mucus layer disappears, like a healthy mucus layer you have, that disappears, and then sometimes this mucus, the epithelial tight junction called become leaky. You heard about leaky gut. That condition could be very detrimental, and a lot of microorganisms grow and then can pass through this barrier very easily, and that's called dysbiotic condition. Okay, and in order to understand what happens, the microbial community forming biofilm, and that really try to maintain the eubiotic conditions or eubiosis. Okay, so if you look at the biofilm formation in the gut, again, remember this morning I keep telling you, no matter what surface, abiotic or biotic, the mechanism is very simple or similar. Okay, so they also start same way on the mucus layer. These are epithelial layer. This is the mucus layer. This is the lumen. So you have planktonic cells, right? They find a group, they form a microcolony, and eventually they mature, and then they disperse, right? And so this biofilm formation is pretty similar as what we talked this morning, right? And then it's dispersed, and then can start again. And so similar thing happening in the gut, okay, when they're forming biofilm. And that's how a lot of our microbial community maintained in a positive way in the gut. So don't think that it is a negative, unless you have microorganisms which are causing severe damage to your gut. And that's the dysbiotic condition diseases. So this is a natural or normal process, right? Let's look at on the left, you have a nice thick mucus layer. That's the healthy conditions, right? Presence of commensal microorganisms on the left. And so this is a mucus layer. These are the microbiota. Okay. What happens when your mucus layer becomes thin or thinner and disappears? When do you lose your mucus layer? Anybody? At what conditions people lose their mucus layer? Because mucus layer is very thick, it maintains the health of the gut. Okay? And that's where a lot of this microbial community reside, and they maintain a healthy environment, gut environment. Okay? I think you raised a hand. You want to answer? Okay, so the answer he's saying, when someone get infected by Helicobacter pylori, the infectious agent, that can also affect your mucus layer, right? Any other conditions? Excessive? Oh, hydrochloric acid secretion. Okay, that can also affect it, but it could be that you have mucus layer damage. That's why you got more HCl secreted. So it's a reverse order. Any other? Anybody else this side? He represented left side. Oh, okay. pH. Oh, so changes in the gut pH. Okay. Okay. Anybody else? So, for example, if you are starving for a couple of days or 2 days or half a day, there is no food for you. When you go back to the hostel today, there is no dinner for you. Then we decided that, okay, there is also no food tomorrow morning. What will happen to your gut? What do you feel? Come on, you need to wake up. I know it's after lunch. Usually it's pretty sleepy, right? I know all of you feeling it. So what did I ask you? So if there is starvation, right? That means you did not eat. So what's your microbe going to do in your gut? They're still there, right? They need food. What are they going to do? They are all going to eat your mucus, right? Mucus is a good food. That's what this microbial community on the left. Okay, so gradually So the starvation, that's one of the major factors. Nutrition, right? So all of your microbes, they're going to chew up your mucus. So eventually mucus is going to go away, right? So what happens? These microbes or pathogens, if some of they are opportunistic or invasive, they're going to get in and cause problems. And so this is what you see, absence of commensal biofilm. Commensal biofilm forms when you have a nice mucus, and that biofilm is gone. Now I'm talking about healthy, desirable biofilm. Not negative biofilm, right? These are good biofilm. We need them. And if your mucus layer is gone, they are going to form biofilm. And then you are going to missing out all your microbial community, because the diverse microbial community in the biofilm maintain our gut health. That's why we are healthy. There are a lot of conditions, hydrochloric acid, achlorhydria, also stress. If you don't sleep and during exam period, a lot of times you stay up all night, your HCl secretion mess up. A lot of those factors play important role in your gut conditions. One condition is called achlorhydria. Achlorhydria is when your stomach do not secrete enough hydrochloric acid. That makes you more vulnerable to infection, because a lot of pathogens can pass easily and can cause problems. So these are good examples for biofilm to be maintained in your gut to keep you healthy. So biofilm and disorder, GI disorder. So what we are saying is that under gut disorders, a lot of diseases happen. If the biofilm is gone, good biofilm is gone, then all those bad microbes, they're going to colonize, they're going to invade, and they're going to cause inflammation. Are you with me so far? The good people gone, the bad people get in, and they're going to cause havoc, right? And that's what happens. A lot of those diseases, IBS, irritable bowel syndrome, inflammatory bowel disease, Crohn's disease, and here also achlorhydria, and a lot of those diseases happen. And these are the microorganisms which are present in our gut. Normally, they are not doing any damage, but when the mucus layer gone, your good microbial community disappear, and they can be invasive, and that's when they can cause a lot of this problem. Okay, in this slide, what you are seeing looks pretty busy. This is saying the number of natural microflora, and this is the different parts of the colon, and this is what you are seeing, natural microbial community, how it keeps us protected. Okay, as soon as you disrupt them, they're in big trouble. So here example, this one, let me see, this one just showing this community, see these organisms and how they are maintained, and these are the biofilm matrix and the bacterial cells inside. This is actual colonoscopy picture, okay, how the biofilm works. But in the disease conditions, okay, so this picture, these are actually very, again, new studies. And so here they are looking colonoscopy before and after jet washing, okay. And here before you can see how red it is, ileum, and this is in cecum, and they try to say if it's biofilm produced by the harmful pathogens or organisms, you want to clean it up, and they clean it up after washing. And so some disease conditions, they are trying to find out that whether this biofilm produced by these undesirable or pathogenic organisms, can it be washed or removed physically inside the gut. Okay, and so that's the actual implication of the biofilm in causing diseases. So of course the inflammatory bowel disease, I don't want to get too much detail, but the whole idea being is that the microbial biofilm, loaded by undesirable organisms, not by natural organisms. So in this slide actually showing these different types of bacteria, viruses, protozoa, fungi, they are part of those inflammatory bowel disease conditions in the gut. Okay. Also the colorectal cancer and inflammatory bowel disease, these are some of those that how this work together within the gut system. The biofilm and the colorectal cancer associated. If you have a cancerous conditions, right? and then the biofilm produced by undesirable organisms, and that can lead to also cancerous conditions. Okay. So this clearly shows that biofilm in the gut play an important role. In a positive side, it can help you maintain healthy gut conditions, and in the negative side, it can cause serious disease and as well as the cancer. So there are a lot of studies actually in progress on this specific topic. And so I know we are getting close. So I know I will not be able to finish it, but one of the questions we started asking is that biofilm's role in pathogenesis. I already answered some of those questions, but I want to ask a very specific question. If you take a biofilm cells, so for example in a laboratory, you have a petri dish, you are growing bacteria, right, and they are forming biofilm. So one question we wanted to ask, are these biofilm forming cells, which are the sessile cells, right? And if you take a planktonic cells, do they have any difference in the pathogenesis? Are you following me? Because we keep asking, the planktonic cells, they eventually form, right, go through biofilm, eventually make sessile cells, some population become dormant, some become persister, some could be VBNC, right? And now they can come back to planktonic. So you wanted to look at, are those sessile cells, are they pathogenic as planktonic cells? That's the question we want to answer, and I think I will follow up tomorrow. It will be a little bit overlap. So our study would be Listeria monocytogenes as a model pathogen to study biofilm mediated pathogenesis. We actually designed that experiment to ask that question, to answer that question. Okay, I will end it here, and next 2-3 minutes, if you have any questions, go ahead. Can you give him the microphone?
Sir, actually, I have the question about that persister cells. Actually, how are they forming? I mean, how are they actually forming?
How they are forming?
Yes.
That's what I was trying to tell you, is that these cells are responding differently to the stress, right? Okay. And so the toxin antitoxin function.
Yeah, about that system, I didn't quite get that. And toxin antitoxin system.
That's why I'm saying you need some read up more. Okay, let's say the toxin, whenever you say toxin, what does toxin means to you?
Harmful substance.
Toxin, any kind of toxin. Yeah. So as soon as you see toxin, that means its purpose is to damage, right? Yes. Whether it's a membrane or enzyme or protein or any of those stuff. So when the cells undergoing persister phase, the toxin part, one of those enzymes which actually degrade that messenger RNA, and that RNA actually codes for enzyme that may be required for the bacteria to become a healthy cell, living cell. So it can go through division. Okay, but if that enzyme is arrested or inhibited, the cell loses that function, therefore it cannot be metabolically active. You see this one? And so that may be the beginning stage of their going through the persister phase. Okay, means so it's metabolically less active.
So they actually have a negative role in persister cells. Actually have a negative role, you know.
Persister cells from the bacterial side is good because they want to survive. They want to persist in that environment. But from our side, from the health side, it's negative, right? We don't want them to be there. We want to kill them because otherwise, you know, you are treated with antibiotics, and these are not antibiotic resistance, but still maintained. And as soon as you stop giving antibiotics, they flare up. They come back to life. They cause the disease again, which is not desirable, which you don't want. So normally doctors practice, I know besides some of the strategies they gave you, what I mentioned, normally what they do, if they realize that we are creating these persister cells, then they can stop the antibiotic treatment for a while and let them build up again, and then give a higher doses of antibiotics to kill those cells so that we can avoid the persister phase.
So we are actually increasing the effect of antibiotics to kill the persister cells.
Yeah, it starts with the antibiotic. So we are trying to show you the sublethal stress also, right? anytime you are not maintaining that level or concentration, always some population going through this persistent phase. Okay?
Thank you, sir.
Any other questions? No? But bring some questions for tomorrow. This is my task. So we'll have plenty of time right before you start lecture or during the break, right? Ask. I want to hear some questions from you. You cannot stay silent. You already participated this morning. You told us your hobbies, right? Or what you are studying. So we will hold